The overall goal of the following experiment is to produce asymmetric ferrocene-biotin bioconjugates that can be immobilized on a gold surface. This is achieved by synthesis of the ferrocene-biotin bioconjugate on a solid phase resin. As a second step, the bioconjugate is removed from the resin, which allows for isolation and characterization of the compound. Next, the bioconjugate is incubated with a gold surface in order to allow adhesion of the thiolates with the gold. The results show that a ferrocene bioconjugate can be produced in good yield and purity and then immobilized on a gold surface.
The main advantage of using this technique over existing methods, like solution-based methods, is that purification is simple and straightforward, in addition to avoiding byproducts produced using solution-based methods.
So we first had the idea of this method when we thought about the efficiency and practicality of the glucose sensors that are used to monitor diabetic patients.
Demonstrating this procedure will be Rodich from my lab and Paulina Gonzalez, undergraduate and graduate students respectively from my laboratory.
First, place 250 milligrams of biotin-loaded resin into a fritted syringe. Swell the resin by drawing up 5 milliliters of dimethylformamide and shaking the syringe on a lab shaker for 20 minutes. When finished, expel the solution and repeat the dimethylformamide swelling. Following this, remove the F-MOC protecting group by adding 4 to 6 milliliters of 20% piperidine in dimethylformamide to the syringe.